Thank you. It's a great pleasure to be here again. I'm not going to give you an easy talk. It's going to be rather complicated and demanding. But you have to get used to this idea, because Kaqun is water and something more. And I'm going to give you some details about this more. What would be the more about? Okay, the presentation is named Kaqun induces gene transcription modification. So I'm going to talk to you about what Kaqun does at the level of our cells, especially at the level of genes. I'm pretty sure that each of you are quite familiar with the idea that whatever is going on in our bodies is driven by genes. So if the gene expression is changing, that has a meaning for the cell, and is going to initiate, gene expressions are going to initiate all kind of reactions, started from antioxidant defense, starting from immune response, and so on and so on. So what we are doing at the moment, we are studying Drosophilas. Why Drosophilas? Because Drosophilas are the best known organisms regarding how they are working, how life is regulated at the level of a multicellular organism. It's the best known organism. Couple of years ago, actually 3 years ago, because we were talking about the Nobel Prizes, 3 years ago the Nobel Prize for Medicine went to a Drosophilist for the finding out how the genes are regulating the innate immune response. The same system is working also in humans, but I'm not going to talk this time about the immune system in Drosophila, probably next year. This is the idea, the new paradigm of life. So basically whatever is going on in our bodies is controlled by the interplay between our genomes and environmental factors. Nutrition is absolutely important. We are very much studying how nutrition, in collaboration with Kaqun, oxygen availability, is interacting in order to sustain life at the level of cells in Drosophila. Also, to have a full picture of what would be important, how to enlarge this picture, we should put in here in this row also the physical activity. But we do not have methods to assess this the moment in the case of fruit flies. This is a picture showing, so what we are going to do today, we are going to focus on metabolism, how metabolism is regulated, and when we are administering Kaqun water to flies, how is going to change their metabolism. Yeah? Actually, if we can provide more evidences that the genes are changing their expression, that would be the ultimate proof that Kaqun is really efficient and is not a kind of fairy tale story. Okay? So again, Drosophila. Why to praise Drosophila? Because you see, there are many kind of metabolic profiles during the life cycle of the Drosophila. We are going to focus today only on what is going on during these larval stages, when basically their metabolism is about, in some parts of the larval body, their metabolism is about aerobic glycolysis, is going up, and when it's going up, basically the carbohydrates are not going to be burned to produce energy, but to produce biomass. This is synthesize amino acids and to synthesize fatty acids and so other things. When it comes about the lipids, the lipolysis and beta oxidation, so burning the fat to produce energy, is going down. This kind of metabolic activity. Why? Because the fat are going to be stored, because right after the larval stage is coming the pupal one, and during the pupal period, basically the animals are not going to do anything. They just going to retrieve some oxygen. Some parts of the body are going to be totally broken down, while the other parts are going to develop into the organs of the fully matured flies. So a lot of metabolic things are going on. So it's an ideal object to study regarding how metabolic profile is regulated. Last year I was telling you about a kind of experiment that we have done. So basically, it's all about viability of the individuals, playing with nutrient availability and oxygen availability, including Kaqun in the story. So this was a kind of experiment when we had raised the larvae on a normal sugar media. This is a kind of media which would support the normal development of the fruit fly. I have to tell you that fruit fly, Drosophila, is the first organism for which the diet and every single component of the diet has been analyzed, and the importance of the diet, of these diet elements, has been defined. So 40 people, 8 years have worked basically in the city, in the Linda Partridge lab. So we know exactly when to administer, for instance, C vitamin, how much to administer to have the development going on in a normal way. So when we were raising the larvae on a normal sugar and normal water, basically, and we applied hypoxia, basically only 22% of the larvae survived. When we basically introduced in this game a high sugar media, a media that contains a tremendous amount of sugar, in these conditions diabetes is going to develop in flies. Basically, due to the diabetes, and we had normal water, only 10% survived, from 22 to 10. Now we were introducing in the game, in the picture, basically the Kaqun water, and I have to tell you that we are also applying hypoxia during the larval stages. So we went down to 1% oxygen, and you can see very few of them survive. But when we were adding Kaqun water into this normal sugar media, basically 84 were getting developed, and with Kaqun 72%. This was a kind of indication that something is going on, so we should get more into details, and we decided to carry out a kind of microarray type of studies. Microarrays is a kind of molecular genetic method to analyze all the genes in the body, how they are working, how the expression of the genes is actually are changing. And basically, we got the results. Actually, exactly tomorrow is going to be a year when we were having this meeting in here, and I was telling you about the results that they arrived, but I couldn't have a look on those results. Now we have the summary of these experiments, what we could see. So in here you can see these lines are representing the expression pattern of all the genes in Drosophila, in larvae that were treated in different ways. And yeah, let me have a bit of water. And the expression level that was increasing is shown in this area, while those that were decreasing are shown in this area. So basically, on the log2 scale are shown the expression levels. So the expression that is above 1, that would represent a twofold increase in the expression. Those that are below -1, a twofold decrease. Yeah? In here we would have not that many genes in this area. In here again, basically in total there are about 1,500 genes, okay, increasing and decreasing their expression levels. So don't leave with the idea that Kaqun, once we administer Kaqun, is going to do only one thing to our body, okay, to our cells. Many things are going to happen, and the expression level of the genes are telling us And certainly we would need to group these kind of genes regarding their functions, but we get there immediately. So what you can see also in here is that, so what were the experiments? So we had Kaqun normal water combination on a normal media, normoxic condition. That's the blue line, yeah? It's going in here. Saying that this would be the expression pattern of the genes. Then we changed the media, so the same setup. From normal media we went to high sugar, normoxia, the green line. You can see the expression level is getting changed. So there is a difference between the blue and the green line. Then we went and we did an experiment where basically we were applying hypoxia. Okay, Kaqun normal combination, normal media. Look at the red line, yeah, and it's quite interesting that the blue and the red line are quite close to each other. So what is the blue and the red? You can see in here Kaqun normal water, normal media, normal media. In here we have normoxia and we have hypoxia. So basically the two lines coming close to each other are suggesting that the Kaqun can cover for the hypoxia, for the hypoxic shock. And the same thing in here for the green and the purple line. Yeah, basically in high sugar conditions, again, the Kaqun can cover for the gene expression pattern that we can see in normal circumstances. These kind of experiments are suggesting that normal media would be the normal diet, high sugar media would be the diabetic type of nutritional setup. And in both circumstances, Kaqun can cover for hypoxia. We were very pleased with these experiments, but these are basically the first clear observations indicating that Kaqun can deliver oxygen to the system, which would be the cell. Another experiment, a bit more complicated, but I would just ask you to focus on the so-called purple line and green line. So what is purple and blue, the Kaqun color, yeah? The Kaqun, the blue line would stand for high sugar, normal media, normal water type of experiment. So when everything is okay, you don't eat a lot, just normal feeding. And then the purple line would stand for high sugar, normal media, Kaqun water, and hypoxia. And you see basically again the Kaqun water can cover for the hypoxia that is induced upon high sugar normal sugar interplay, suggesting that basically it's the same thing in other ways. Look, the purple and the blue coming together. There are some differences regarding how many genes are implicating in these kind of responses. That is shown in here. That's shown in here, and the logic behind this kind of number of gene expressed, I mean with different kind of expression levels, are indicating also a kind of implication of the Kaqun in scaling down the harmful effects of the high sugar load. So having seen this kind of data, we were really happy, and believe me, I'm absolutely happy with these kind of things, with these kind of results. So we said if so many genes expression is going to change, right upper Kaqun treatment, whether in normal diet or high sugar diet, whether hypoxia is around or not, then let's see what are those genes doing. And Drosophila is the organism where basically the majority of the genes have their functions defined. So basically from this point of view, Drosophila is the best known organism. So we were focusing only for the moment on the genes that would stick out regarding their expression levels. So that would be in the so-called high value and low value region. And basically we were looking at the function of these genes. Yeah, and it turned out that some of them are implicated in amino acid metabolism, lipid metabolism, carbohydrate metabolism, so actually in the metabolism. Others are implicated in the immune response, hemolymph hormone type of issues, Then the cuticle and the chitin. This is again a kind of function in the flies, which is related partly to carbohydrate metabolism and immune response. Mitochondria and cytoskeleton genes are also implicated. Signal transduction, cell cycle regulation, translation, transcription. So all these elements, which would be about the communication inside the cells. Proteolysis or protein degradation. Oxidoreductases, which are very important for the regulation of the redox status of the cell. So dealing with stress, oxidative stress. There are also some master genes, very, very important genes, not that many, and there is another class called unknown genes. So where to start now analyzing these kind of functions, and how these genes, through their function, how they can contribute in delivering delivering oxygen and making the cell to cope with nutritional stress was the question. But prior to that, here you can we were applying a kind of statistical analysis called principal component analysis, where all the members of these 20 functional gene groups have been included, and the color codes would stand for every single gene functional cluster, and you can see some of the genes sticking out from this representation. So these would be the most relevant genes to start with and to analyze, because these are the genes that do have we believe we do have a really serious implication in what is going on when we do normal type of diet or high sugar diet, and then hypoxia is around or not. And you can see, just for the to pick up some, is the actin genes and actin 5 and then myosin type of genes. So genes that are implicated in the actin-myosin network, or complexes which are about muscle type of functions, suggesting that Kaqun does something to what is going on in our muscle cells. Yeah, there are other genes in here, like LCP1, 3, or LSP. These are implicated in the transport of the nutrients inside the hemolymph or the blood, while many others are clustering in here. But let me give you some more details of what is really going on, because time is really the actin genes. Yeah, so we have actin 57B and actin 5C. Just to understand the experimental setup. So this would be a normal sugar, normal water type of condition. This would be a normal sugar, Kaqun water type of setup. This is a kind of normal sugar, normal water, but applying the hypoxic shock. This would be normal sugar, Kaqun water, and applying hypoxia. And these four are the same, but the only difference to these four would be that these four are with normal sugar type of media. These are with high sugar media. So we were combining the nutrient availability with hypoxia, and also with the water that we were administering to the flies. This would be 167, stands for the normal type of gene expression, at least for actin 57B. If we are going to apply, we replace the normal water with Kaqun water, you see the expression of this gene is going up. So why is gonna increase? Because it is needed, we presume. Then if we apply hypoxia, basically you see it's gonna go back, it's gonna fall back, the expression of the gene. Again, if we apply Kaqun water, it's gonna decrease. Certainly we would expect to have a situation when it's gonna increase, but it's not happening. But then if we go to the experiment where basically, we replace the normal media with high sugar, you see comparing this to that, you can see an increase, a really serious increase. When we replace the normal water in a high sugar media with Kaqun water, we have even a higher increase. Then we apply hypoxia, is going to go even higher. Then we apply hypoxia and replace the normal water with Kaqun water, and basically you see the expression of the gene is going to fall back. So don't get the idea that you can stretch the system as much as you can. So we can do many things to our organisms, but we cannot stretch it until you think it's possible. There are limits, and when we go over certain limits, it's gonna fire. It's gonna fire back. So there is the so-called effect and contra-effect type of interactions. And Kaqun is acting, is doing something to this system. Sometimes it's stretching, sometimes it's gonna get back to normal. Yeah, here too. So in the case of the other gene, when we have Kaqun water in the normal media, basically it's gonna fall back the expression. But if we apply actually a hypoxia, is going even down. I know it's pretty complicated. It's really complicated, believe me, but we are not going to lose our patience. I know you are right at the border to losing your patience and becoming fed up with these kind of things. This is what scientists are doing, okay? It's a matter of finding out what is this interplay all about, okay? What is this interplay all about? So in other terms, what is really happening in here? So if we apply a lot of sugar to any kind of animal system, then basically we can have a situation when a lot of fatty acid is going to be produced, and then basically with that fatty acid we can do other things. We are moving now to biochemistry, and very soon it's going to be a combination of biochemistry and genetics. So you will be absolutely fed up right after this presentation with these kind of things, and you would say how lucky I am that I'm totally parallel with these kind of things. Okay, and only lunatic people can do such type of science. Hopefully it's gonna lead us to interesting things. Okay, so in other words, if we eat a lot of sugar, then in Drosophila and also in humans, this kind of signaling system is gonna kick in and is gonna give some orders to the cells. So and again, the signs in here are representing the earlier mentioned experiments. Light blue stands for a situation when we were applying normal diet and Kaqun water, and the dark brown stands for a high sugar media, and the dark blue stands for the situation for high sugar and hypoxia type of combination. So you see, this is a kind of transcription factor that is going to tell the cell what to do with the sugar that's around, and basically the equal sign in here is telling us that if you eat normally and you drink Kaqun water, is not going to give any signal to the cell, the system, that you must do something. I mean, you have to do something different to what you were supposed to do. But if you eat a lot of sugar, basically this system, the Mondo system, is going to tell the cell, okay, you have to do something with this sugar. And through these kind of signaling systems, it can direct the events to lipogenesis, for instance. That is really happening, and especially in the case of Kaqun, with high sugar and Kaqun case, or through the Kabut system, we can reset our circadian clock. 4 years ago, the Nobel Prize for Medicine also went for the circadian clock. Again, Drosophila was implicated. Okay, and so on. So we know a lot what is really going on, and you see Kaqun either increases certain aspects through these kind of genes, or is going to decrease. Yeah, and then we move to, because it's about metabolism, and mitochondria are heavily implicated in metabolism. Starts with the glucose and how the glucose is going to get to acetyl coenzyme A, and then how that one is going to get introduced in the Krebs cycle. We Hungarians, we tend to add Krebs and Szent-Györgyi, Albert Szent-Györgyi cycle. You know, the guy who discovered the, we are saying, we are exaggerating because we are Hungarians, okay, who discovered the C vitamin C, the C vitamin, in the red paprika. That's why Hungarians are called Paprika Nation. Okay, good. And then how the NADs and the fats would flow to the ITC in order to obtain ATP to cover our energetic needs. Here it is, the whole process in more details, suggesting this is a very important point in all this ATP production in the cell. And basically, what is really happening, it appears also a kind of lactate and also the acetyl coenzyme A. That's quite strange to have these two guys together, because actually the Nobel Prize for hypoxia type of regulation for the HIF1 implication. HIF1 is implicated in regulating these kind of events too. But in here, it's a situation when both lactate and acetyl coenzyme A is going to get produced. That's very strange. We do not really know the reason for this, because hypoxia is there. In hypoxic conditions, actually, the HIF1 is there to separate, to make a clear distinction between this pathway or that pathway. Yeah, but we'll sort this out. So once again, I just want to emphasize that Kaqun has an effect and is in regulating these kind of events, and also in here these pathways that are also related to glycolysis are not really favored in the presence of Kaqun. Even more detailed biochemistry. That's the reason you see these complicated issues. That's the reason why medical students, biology students, not really adore biochemistry, because there are so many things in here. Again, you see in blue, you can see the enzymes implicated in the Krebs cycle, and what it turned out is basically a situation when the alpha-ketoglutarate is going to be turned away from the cycle, and when we look, and it's going to be turned away to obtain glutamate, and then from the glutamate to obtain GABA. You know this neurotransmitter, very important one. This kind of pathway is called the GABA shunt. Is really important, and Kaqun is very much in the favor of producing GABA. And why we were really happy to see this, because the GABA is implicated in relieving anxiety, pain, regulating sex hormones, ADHD, burning fat, stabilizing blood pressure, blood sugar level in the case of diabetics. So if somebody is asking what would be the take-home message of all this stuff? The take-home message of this stuff would be that if this is really the case, so Kaqun is capable of increasing the GABA synthesis, then we are in the right place to try to study the Kaqun's effects on diabetes. And not just diabetes, but also think about ADHD, or think about the PTSD. Very interesting possibilities ahead of us. So we are basically providing some links to what exactly we should look at, what patients we should assess regarding the Kaqun water's effect. Yes.
Yes. Sorry, but we all see this in the clinicum. We all see this.
Yes.
We all see this effect.
Yeah, exactly. I know the story, but I can tell you that this is the reason. This is the reason, because basically through this biochemical pathway, This is basically the foundation of what you were seeing in the clinics. Yeah, it's based on the GABA. So Kaqun is favoring the GABA production. Not talking how interesting would be this to try in the case of people suffering of depression. Okay, which is another.
We have studied on that.
Good.
That's the oncology hospital.
Yes.
Very interesting. And it was the result was excellent.
Yeah. Now we have the foundation, the scientific foundation for the effects that you have seen in the clinics. So nobody can say that this is a kind of, you know, whatever reason for that. Yeah, it's the GABA.
So all I have to learn GABA now to explain.
Yes, yes. That's the next story.
Can I
Yeah.
sign up in your course?
Yes. More to come, but now we move a little bit to the lipids. So here you can see basically how the sugar enters the fat cells in Drosophila and how it's going to favor the production of fatty acids explained through all this system. So when basically this enzyme is not really fully efficient, a lot of methyl glyoxyls are going to be produced, and that would lead to H type of products, and also initially to insulin resistance. Okay, what we see in here is that due to the Kaqun is going to increase the expression level of this gene, so which would favor the production of fatty acids through fatty acid synthesis. Again, you know, the expression is high in the case of Kaqun, suggesting that basically a lot of sugar is going to get transformed to fatty acids. That's okay, but there will be no additional products in order to lead to aging and also insulin resistance. There is another gene implicated in the detoxification of the methyl glyoxyl, this one, GLO1, and that expression too is going to increase. So Kaqun is doing two great things with a lot of sugar in us. If we eat a lot of sugar, it's gonna lead to a lot of fatty acids, and there will be no aging, insulin resistance, and other toxic substances around. So it's gonna help also to the detoxification. That's again amazing. Because insulin resistance is another complication that we have to fight back, and we can fight back insulin resistance in many planes, in many directions. This is one of them, and actually Kaqun is really is going to be a good tool to look into how efficient is in this stuff. Let me skip this, talking about lipid metabolism. Okay, I'm not going to explain you all in details because we would need at least a few more hours to that, but just look at the colors. Again, just look at the colors. This is the insulin pathway, you see, and then this is another type of pathway, the adipokinetic type of pathway, and other elements. I would just pinpoint the idea that the fatty acid synthesis is favored very much by the Kaqun. Whenever we are overeating or we eat a lot of carbs, which is great. But the whole scheme is telling us that Kaqun is not very much in the favor of putting away the fatty acids in the so-called lipid droplets. It's very much about synthesizing a lot of fatty acid, and then we should do something with this fatty acid, and this is about if we can do something. What we do with the fatty acids to take it to the mitochondria, and through the beta oxidation to get ATP. However, in this respect, Kaqun is not very much in the favor of sending the fatty acids to the mitochondria, carrying through the beta oxidation, because this enzyme activity is very much inhibited through Kaqun. So we have to define what exactly is happening with the fatty acids that are produced in the case of carbs, overeating carbs. Yeah? They are not going to be put aside, but then what else? This is also a scheme about explaining how the hormonal regulation is going on in Drosophila. Pretty much the same elements are around. I would just pinpoint this type of element in here, the NLES. This is a gene that is in humans is implicated in insulin resistance. Okay, so if this gene gets expressed at high level, basically it's going to lead to insulin resistance. In the case of if you are when the flies are eating normally and they are administered Kaqun water, you see the expression of this gene is basically decreasing. So that's a good news. That's a good news. That's something that we would expect, but it's a good news, suggesting that Kaqun is decreasing the expression of this gene and is fighting back the insulin resistance. This is relevant for diabetic people. Why? Because diabetic people on a normal diet still have diabetes, and we don't know why. We know why, but the side effects are there, and the insulin resistance is there. So giving them normal Kaqun water is gonna decrease the insulin resistance. Okay, just one such a message. Let's skip it. And what else is doing us the Kaqun? We are inside the mitochondria, and it was quite a surprise for us seeing that the ITC electron transport chain activity is going high, but there are no reactive oxygen species produced. Why? Because all these enzymes, the superoxidismutases, the glutathione peroxidases or catalases outside the mitochondria, the expression or the mitopherins expression is going to stay the same. So this is a really sensible system. So if a lot of reactive oxygen species are produced immediately, the expression are going to go high. So in other words, when we are administering Kaqun, it seems that there will be we should not expect a lot of reactive oxygen species to be around or to persist for a very long period of time. So the system would kick in and would keep push it down very efficiently. But the system is really high and is going to produce a lot of ATP. So basically, what would be a kind of take-home message? The take-home message would be regarding mitochondria that whenever we have Kaqun around, there might be some reactive oxygen species, but shortly afterwards are going to get this stabilized, and then a lot of ATP is going to be produced, and this ATP on the cellular level, 80% of the ATP is used for translation for protein synthesis, and also to keep alive the sodium potassium ATPases in order to maintain the proper polarity of the cells. So we are going to have a lot of energy, and that can be used to sustain the normal functioning of the cells, getting close to the final, to the finish. So we have seen multiple effects during administering Kaqun, and these kind of effects are very much related to many functions, and we are very much into finding out the interplay, how these kind of functions do collaborate in order to maintain the proper functioning of the body. And also it's pretty obvious that some effects of the Kaqun depend very much on the nutrient and oxygen availability, and also Kaqun can cover for the hypoxia what would be the normal functioning, the normal expression of the genes in flies. And thank you for your attention. I can promise you that next year, if you are going to invite me and you would say, don't come because you just make these people to go away, to turn away from Kaqun, because it's getting so complicated, all this story. Yeah, that's exactly how it's happening. Initially, everything looks very simple, then things would complicate, and then eventually things will settle, and then everything is going to be again simple. So now we are moving to what would be the complicated stage of the Kaqun story. Okay, thank you very much.
Thank you.